So welcome everybody to the May edition of the Monash Addiction Research Centre Interdisciplinary Webinar Series. Before we get going, I just want to acknowledge the traditional custodians of the various lands on which we work and meet today, and I pay my respects to elders past, present and future. So the Monash Addiction Research Centre Interdisciplinary Webinar Series aims to bring together addiction researchers, clinicians and other experts from the sector to discuss emerging research and challenges in the addiction field. I'm excited about today's topic. Today we're going to be talking about obesity and the cognitive and neural mechanisms of compulsive eating. So we have three fantastic speakers who I'll introduce in a moment. I do just want to remind everybody as we go today, we really look forward to having a great discussion at the end. And so if you have questions that occur to you along the way, it would be great if you could put those into the Q&A box as we go, so you don't forget your excellent questions. Not to use the chat box and really focus on the Q&A box so that we do see all those questions and we're able to cover them at the end. So we have three fantastic speakers. Our first speaker today is going to be Dr. Katharina Voigt, a postdoctoral research fellow at the Turner Institute for Brain and Mental Health. Katharina's research focuses on the understanding of brain processes that underlie our preferences and choices using advanced neuroimaging methodologies. So she received a degree in psychology and a master's in cognitive neuroscience in Berlin. Before moving to gain extensive training in decision neuroscience at research laboratories across Europe. She has a PhD in cognitive neurosciences from Melbourne University, and during her PhD, she investigated the neuronal and cognitive underpinnings of food choices. And during her postdoctoral training, she's further advanced this research in decisional neuroscience in the field of obesity and also using a range of neuroimaging methodologies. Our second speaker is Professor Zane Andrews, who is the head of neural control of energy homeostasis lab at the Monash Biomedicine Discovery Institute. Zane is an NHMRC Senior Research Fellow and is internationally recognised for his work on the neuroendocrine control of energy homeostasis and behaviour. Professor Andrews uses preclinical animal models and viral genetic techniques to study how the brain controls food intake and feeding behaviour, and this includes the role of homeostatic and hedonic systems and how they interact to influence both the need and the desire to eat. And his lab is particularly interested in the why and the how of how the brain promotes the overconsumption of highly palatable, energy-dense foods and how this contributes to obesity. And we are discussing today, we're very pleased to welcome Professor Antonio Verdejo-Garcia, who is the Deputy Lead of Addiction and Mental Health Program at the Turner Institute for Brain and Mental Health. Antonio is a professor and a Medical Research Future Fund Fellow at the Turner Institute. He has a PhD in psychology from Granada in Spain. You can see it's a very well-travelled panel that we've got today. And then he has done his postdoctoral training in addiction neuroscience at the University of Cambridge in the UK and neurology at Johns Hopkins in the US. And so his research focuses on understanding brain mechanisms and thinking processes that drive addictions, overeating and obesity. So without any further ado, I'd like to hand over to Dr. Voigt, who is our first speaker.
Okay, I'm just sharing my slides. Can you see that okay?
Perfect.
Yes, thanks, Susie, for the introduction and the invitation, of course. I would like to hit off the session by presenting some of my recent work from my postdoc at Monash University at the Addiction and Mental Health Lab, during which I looked at the communication of brain areas that usually guide our food choices, and then how that communication might be disrupted in individuals with obesity. So let's start off with a few statistics. So obesity is actually one of the most prevalent health problems worldwide. So starting from only 1975, within only 40 years, the frequency of obesity actually more than doubled. And now we live in a world where there's 1/3 of the population considered as overweight or as obese. And we are living in a world where there are more people overweight than underweight. So in brief, obesity is a global epidemic. And if we look into the numbers of Australia, they're even more shocking. So for example, in 2018, 67% of the Australian adults were actually considered as overweight or obese. And this number actually doesn't stop here. It continues to rise at an alarming level. And that's a problem because obesity constitutes a burden for the individual. For example, it increases the risk for cardiovascular diseases, medical conditions such as diabetes or certain cancers. But also, of course, it has a high impact on the quality of life for the individual, as you probably can imagine. And it can shorten the life expectancy by around a decade. And on top of that, obesity also has a large economic burden. So for example, in Australia alone, the combined economic impact is estimated to be at around 60 billion annually. And so I hope that these numbers make it clear that understanding the driving factors of obesity is actually critical if we want to manage that national and international health crisis. And that's why it's a priority on current research agendas such as ours. But of course, that's not an easy task, as obesity is a complex condition. And if it were also easy, we would have solved that issue by now. And as psychologists or neuroscientists, we are mostly trying to understand diet-related or choice-related causes of obesity. So from that perspective, to break it down to the most simplest driving factor, obesity is actually caused by food overconsumption. And food choice itself is another complex process, and it's driven by a large-scale brain network, which I briefly summarized here. And in the center or a core area of that network is actually the human hypothalamus. So the hypothalamus is a well-known center that regulates feeding based on our homeostatic needs, such as hunger or satiety. So for example, when we're hungry, the hunger hormone ghrelin acts on the hypothalamus, which then initiates food seeking. And I guess we hear more about that later on in Zane's talk. However, now we live in an environment where we are constantly bombarded with food cues, and we see fast food chains on every corner in Melbourne or in Australia. So our food choices cannot be simply driven by the fact that we are hungry or sated. So there are other systems such as the reward system. They process whether we like something or not. So they help us to decide which foods we like or which we don't like. Then there are systems such as the cognitive control system. So as they assist us to favor long-term goals over short-term rewards. So for example, if I have the goal now to lose some weight, and that's a long-term goal, but I'm confronted with a donut or an apple, that's a short-term reward, that system would help me to actually choose the apple over the donut. And then finally, there are also systems involved in sensing our bodily states. So these help us for correctly interpreting when I'm full, when it's enough, or when I'm actually hungry and I need more food. So understanding this dynamic causal interaction between these systems is actually critical if we want to understand food choice in general, but also, of course, in obesity. And indeed, there are a few theories already that summarize how choice-related obesity results from this disruptive interaction of these systems. So for example, the hedonic eating view assumes that there's an altered communication between the homeostatic system and the reward system. So for example, in that view, in line with that view is that individuals with obesity have a higher food reward sensitivity that override homeostatic needs. Then another view is the eating without hunger view, which highlights the miscommunication between systems that encode interoception or homeostatic regulation. And here, previous studies linked lower interoceptive awareness, so the sensing of whether we are full or hungry, for example. So they linked that to disruptive connectivity between the hypothalamus and the insula. And then finally, we have the self-regulation view of obesity, which emphasizes an impaired top-down regulation of lower striatal and limbic regions. And in support of that view, previous studies found that connectivity in prefrontal regions, so when that was higher, this was an indicator whether someone was successful in dieting or not. However, how these brain areas interact, or these systems in individuals with obesity, and also how it's modulated by homeostatic state, has not been studied within one unified account. And this is actually critical if we want to understand, or if we want to actually develop new approaches to prevent and treat obesity. And so this is where our study comes into play. So we aim to investigate the communication among the food choice networks and how this works in obese and in healthy individuals, and then how that communication is further impacted by our homeostatic state. And we use that by using resting state fMRI and a novel computational method. So just briefly to the method, so just in case some people might not be familiar with neuroimaging method. So resting state fMRI is actually a very simple and fast, but very powerful method. So it works that we put participants into the scanner. We ask them to just relax, don't think of anything in particular, and to fixate a central fixation cross on the screen. And this has been shown to actually be a valuable tool to reveal these underlying brain network patterns. So we use that method in order to obtain our food choice networks. And then we adopted computational neuromodeling on that network in order to look how these areas communicate with each other. And again, how this is modulated by BMI and by homeostasis. Okay, so we had 41 participants in our study. From those, 20 had healthy weight and 21 were obese or had excess weight. And they completed 2 scans, one after an overnight fast, so that's our fasted condition, and one after a standard breakfast, which is our sated condition. And the scans were around 1 week apart. And then after they completed that resting state scan, participants also were asked to do another task in the scanner, which we call the food choice task. And here in that task, here on the slides, you see an example trial. Participants were asked to choose between healthy and unhealthy drinks. And the drink they actually chose was delivered to them via a scanner-compatible gastonometer. And we used this task-based activation in order to find the location of the food choice network within each individual participant. So once we had that food choice network for each individual participant, we then applied our advanced cognitive computational method, which allows us to understand how the areas communicate within each participant. So first we estimated fully connected BCM for each participant in each condition. And then we used Bayesian model reduction procedures that helped us to actually find the best fitting model for each participant and each condition. And once we found that, we looked in the group level and we investigated our main questions. So we looked at 3 effects here. We looked at the main effect of homeostasis. So how does hunger over satiety interrupt the communication of our food choice network? Then what was the main effect of BMI? So with increasing BMI, how does this affect our food choice network? And then we looked at the interaction between these 2. Let's jump to the results. So let me just briefly explain what these figures mean here. So the plus and negative sign you see in these figures mean the variance of a connectivity. So they can either be excitatory. So that means one area can actually increase activity in another area, or they can be inhibitory. So they can decrease the activity in another area. And green or red arrows mean that there's a decrease or increase in that connectivity. So red is decrease and green is increase. So for example, here you see that there's a increase in the excitation from the VMPFC to the ventral striatum. All right, so first we look at the main effect of homeostatic state. So how does the food choice network communication change when we're hungry as opposed to sated, and that's irrespective of weight status. And we see that during hunger, the ventral medial prefrontal cortex, which is implicated in coding food preferences or like value for food, excited activity in the ventral striatum and the hypothalamus, which are implicated in reward seeking and energy regulation. So that means when we're hungry, the brain activates mechanisms that increases the value for food, and so that we actually go out and seek food, which makes sense. And given that we found this across weight status, so it didn't matter whether someone had excess weight or healthy weight, this mechanism was across the individuals. So we interpreted this as a general adaptive mechanism. And so this was consistent with the hedonic eating framework, and we actually can rule out that this is a driver of obesity or a sole driver. Then if you take now BMI into the equation, the critical finding here was that there was a decreased excitation from the dorsolateral prefrontal cortex and the ventral medial prefrontal cortex. And this connectivity, so especially higher connectivity between these two areas, was previously associated with successful dieting. So individuals who were successful dieters actually had a higher connectivity. So now here we find actually this connectivity is decreased in individuals with obesity. And we interpreted this that this might explain the difficulty for controlled food intake, especially when they're hungry. And this is consistent with the self-control framework. And then finally, if we look into the interaction, we find that individuals with higher BMI had an increased excitation from science coding regions to energy regulation regions such as the hypothalamus. And this finding might explain why individuals with excess weight might have a hypersensitivity to internal lipolytic states that can override homeostatic metabolic needs, which then indeed leads to food overconsumption. And this finding is consistent with the eating without hunger framework. All right, so just to summarize the set of complex results. So although what I've mentioned before, increased reward sensitivity has been a prominent account of obesity, we cannot really see that in our data. And rather, our results suggest a model in which obesity is driven by disruptive self-regulation during energy depletion, so hunger, and increased science coding that override the processes of the hypothalamus across hunger and satiety. So what can we learn from this, and how could we use this to improve existing treatment programs for obesity? So our results actually highlight the potential for intrinsic predispositions or neuromarkers, which might inform the development of neuroimaging-based treatment approaches. So for example, we could use that to develop individualized non-invasive brain intervention methods, such as brain stimulation or neurofeedback, in order to rewire the cortical subcortical network abnormalities we found here in obesity. And then to take it actually a step further, so what I presented today is group level data, but you could also do that for each individual and then really look into what is the driving connection here that drives this behavior in that specific individual, and then really individualize it for that person. And similarly, by revealing these individual brain behavioral patterns, this method also offers the opportunity for develop subject-specific treatment programs for obesity. So as I've tried to emphasize, there are these different mechanisms that could drive the disorder, but they might not be unique across, we probably know that, not unique across individuals. So this method actually can detect the specific brain behavior pattern and then target that specifically. All right, that's it from me. Thanks again for the invitation, and of course to my collaborators. Without them, the work wouldn't be possible. And one of them is actually Zane, who will go next. That worked.
Sorry, everybody. Good afternoon, everybody. I was just muted there for a second. I assume you can see my screen.
Yep.
Good afternoon, everybody. It's a pleasure to talk to you today. I'll be talking to you today about some of the work that I do, and that's based on preclinical models, but using information that we can collect from human studies, similar to what Kati was just talking about. Now, it's my, I guess, scientific opinion, similar to what Kati was talking about, that the problem with obesity today that we face is that we are consuming far more calories than we have in the past. And you can see that quite nicely from this diagram here, where daily supply of calories has increased over the last 50 to 60 years. An exception around about the fall of the Soviet Union in Eastern Europe. So that's a very nice set of data. It shows that there are more calories in our world today. And so I feel like the primary cause of obesity is the overeating. And this continues to grow, and it continues to grow in the face of a number of dietary interventions, social interventions, and educational approaches. And so while these are important, I feel it's not getting to the heart of the problem, and the heart of the problem is the underlying biology of why we're driven to overconsume food. And in terms of my opinion, I think the brain is critical here. We know from genome-wide association studies and other genetic studies, this is done by Sir Steve O'Reilly, who is a pioneer in the genetics of obesity. They have concluded that most of the genes discovered thus far have a principal impact on hunger, satiety, and food intake. This was in 2008. And more recently, in 2015, there was another large genome-wide association study. And just to point out, this was conducted on over 300,000 individuals. And again, they concluded that there's strong support for a role of the central nervous system in the predisposition to obesity through the regulation of energy homeostasis, including food intake. So I guess the important thing to think about here is that any approach to treat obesity or overeating in humans should include a fundamental knowledge of how the brain normally controls food intake and feeding behavior. And of course, what I've got here is I'm saying it should be relevant to humans when possible, and we can utilize preclinical models when necessary to test circuits. And I've been working in this field for about 15 years now, and it's fair to say that in terms of the way the brain controls food intake, there's been quite a lot of focus on homeostatic systems, particularly through the hypothalamus. And I guess it's kind of interesting if we break this down, because I've come to the realization that this doesn't explain what's going on in humans. And one of the issues is that homeostasis only really defends against energy deficit. Now, if we think about that in an evolutionary context, you can understand that if you don't eat enough, you eventually starve and die. There is an evolutionary consequence to that. However, there's no consequence to eating too much, or there hasn't been a consequence for eating too much. There potentially as a consequence now, because obesity drives things like infertility. And so into the future, we may see the impact of this evolutionary consequence. But the homeostatic model doesn't essentially explain why humans overeat. And I think we all intrinsically understand that as humans, we eat for a number of different reasons. We eat in social settings. We eat when we see cues that predict food palatability. We are sometimes lazy and take easier options. And so our brains are essentially integrating a whole lot of this environmental information, the sensory information. It's being collected and compared to homeostatic systems. And then we come up with a broad range of different factors that can influence feeding, as you can see here: food intake, mood, choice, all these sorts of things. And of course, it's important to note that these factors can then feed back in the way in which we perceive some of these environmental cues. And so in thinking about how to study food intake and why we want to consume food going forward, we need to leave behind this homeostatic model and essentially have a model that integrates communication between homeostatic systems and non-homeostatic systems. And essentially, what we need to address in order to understand this is we need to be able to identify pathways that are relevant from human data, such as Katie's just talked about. But we need to be able to functionally manipulate these circuits to determine causality, because one of the key limitations of all of the work that Katie talked about is there's no causal evidence to suggest a functional role of those pathways, but we can work that out with preclinical models. And so that's exactly what I'm going to show you now. And this is essentially an extension of what Katie was just talking about. We've been looking at brain connections between hypothalamic regions and non-hypothalamic regions. Katie has used her modeling approaches to predict the directionality and valence of these pathways. And sort of extending on from what Katie was talking about, using this modeling approach, she was able to predict from another set of data that high BMI is associated with decreased inhibitory influence from the prefrontal cortex to the lateral hypothalamus and anterior cingulate cortex to the lateral hypothalamus. And this is what's seen here. Now, this is interesting because this suggests that these pathways would normally restrain feeding and prevent the development of obesity. And so this is a nice prediction that we can now test in preclinical models. And I just wanted to say that we would have never identified this pathway and looked at this pathway had it not been for the information and modeling procedures that Antonio and Katie have conducted here. Also, just to point out, the lateral hypothalamus is important because it's been well known for a number of years that the lateral hypothalamus regulates food intake and reward. Decades ago, people showed that lesions of the lateral hypothalamus reduced feeding and that electrical stimulation of the hypothalamus increased feeding or increased the willingness for rats to work for that electrical stimulation. More recently, we can use optogenetic techniques, new approaches to selectively turn on neurons with light. This is a very cool and exciting approach. It's revolutionized neuroscience over the last 10, 15 years. And in this study here, you can see that activation of GABA neurons in the lateral hypothalamus massively drives the consumption of food and is also rewarding because mice will increase self-stimulation. On the other hand, though, it's been shown that glutamate neurons in the lateral hypothalamus can actually suppress feeding. As you can see here, activation of this pathway reduces food intake. It also causes aversion. And if you ablate this pathway, so if you remove this suppression of food intake, as shown in the red here, you can see that mice will gain weight. So the fact that the human imaging and modeling predicted that the LH was important was nicely in line with existing data. So we wanted to explore the functional consequence of the PFC to LH pathway in mice. And just to put everybody on the same page, I guess, in mice, we can do a number of techniques using genetic approaches to turn on this pathway, to turn off this pathway, to manipulate this pathway, specifically from the PFC to LH. And the way we do that is we use a system called Cre recombinase and LOX-P. And this essentially acts like a genetic lock and key system. You need Cre recombinase, you need Cre present for the system to work. And in this case, we have put a virus that delivers Cre into the LH, but it's retrogradely transported back to the cell bodies. So it goes back to where the neurons originate from, including the PFC. And then we can come along and we can put our Cre-dependent virus into the PFC, and that enables us to control this pathway. So we can turn this pathway on or off as we choose. This is a mouse brain, images of a mouse brain. And all I want to do here is show that we can confirm that this pathway exists in mice. So we put a retrograde tracer in the LH, either a viral approach or a non-viral approach, and you can see neurons labeled in the prefrontal cortex. So there is a monosynaptic pathway going from the prefrontal cortex to the LH in mice. The next thing that we've done is we've artificially turned these neurons on to see what they do. So we've artificially turned on this pathway. Now, in order to do this, we use a system called DREADS. This system stands for Designer Receptors Exclusively Activated by Designer Drugs. And this is a very cool system because it's a modified receptor that has been genetically manipulated so that it doesn't respond to any endogenous ligand. It only responds to an exogenous ligand, CNO, that we can give when we want to. And so therefore we can activate this pathway. And in this case, the first set of experiments, we've put in a stimulatory receptor. So we're going to turn on the pathway from the PFC to the LH and observe what it does. And that's what we've done here. And what was really interesting, we turned this pathway, we activated this pathway, and what it did is it actually suppressed food intake over time. So this is at the beginning of the dark phase, and you can see activating this PFC to LH pathway reduced normal food intake. It also suppressed food intake when mice were fasted and then allowed to refeed. It's not completely reducing food intake, as you can see, but there is a significant suppression of food intake relative to controls. We also then wanted to look at motivation to determine whether or not this pathway influenced motivation to seek out sucrose rewards. And to do that, we developed this mouse vending machine, which we call FED3, so it stands for Feeding Experimental Device. And this is a really cool device because mice have to work to get a pellet. So they have to learn an active nose poke delivers a pellet, and that an inactive nose poke does not. And you can see mice get this. They are trained over time, and they understand that the active poke delivers a reward, a sucrose reward. Now with this, we can then go on and actually test motivation. Oh, actually, here we go. Here's a brief old video that just shows it in work. There's the mouse, comes out, pokes, gets a little sugar reward, and he's happy. And he'll come back for another one, but I don't have time. In order to assess motivation, we use what's called a progressive ratio. And this is where the number of pokes required progressively increases for the same number of rewards. So it would go 1 poke, 1 reward, 3 pokes, 1 reward, 6 pokes, 1 reward, and it will continue going on until the mouse decides to give up. When it gives up, it's called the break point. And this is what we use as an index of motivation. Now, what you can see here is we activated this this PFC to LH pathway with our DREADS. And what we see is that this suppresses motivation because the breakpoint ratio is significantly lower. And it also, as you would expect, suppresses the number of pellets received. So these experiments with activation show that the MPFC pathway that projects to the LH suppresses food intake, but it also reduces motivation to obtain a sucrose reward. We then asked, well, what happens with artificial inhibition? Can we turn off this pathway and then see what happens? And so this is what we did. We used this genetic approach that I told you about before, where we can specifically inhibit only neurons from the PFC to the LH. When we did this and we looked at normal home cage feeding, we found that there was no difference. And this was interesting. So we thought, okay, there's no difference in home cage feeding. So perhaps this pathway is not active and therefore inhibiting it would have no effect. So when would we predict this pathway to be active? Well, there is evidence that the PFC integrates stress, for example. And so what we thought we would do is we would employ a novel environment or novel foods, because mice find both a novel environment and novel foods mildly stressful. And so what we did is we thought we would inhibit this pathway when placed in a novel environment or novel foods in a familiar environment. And what you can see is when we inhibit this pathway and give chow in a novel environment, mice will eat more if we inhibit that pathway. That's pretty cool. We can also see that if we give them palatable rewards, so Fruit Loops in this case, again, mice in a novel environment, if we inhibit this pathway, they will eat more. Finally, it also works if you take novel food in a familiar environment. So here, they're in their home cage, but we give them a novel food. They normally show some level of neophobia to new food, so they won't immediately approach it, unless we inhibit the pathway from the PFC to the LH. So this is kind of cool. It suggests that inhibition of this circuit doesn't affect feeding in the home cage, but promotes feeding in response to stressful contexts. Of course, what I've just shown you is artificially activating a pathway or artificially turning off a pathway, but it doesn't tell us when this pathway is inherently active, when it is physiologically relevant. And so that's what we wanted to do next. And in order to do this, we exploited some other exciting genetic approaches. And this is a genetically encoded calcium indicator. It predicts neural activity by emitting light. And again, we can put this genetically encoded calcium indicator only in PFC neurons projecting to the LH. And actually in mice, we can measure the amount of light given off as it moves around its cage and interacts with objects and other mice and different things. So this is a way in which we can assess neural activity of the PFC to LH pathway in mice that are freely moving and behaving. And this is a really novel and exciting technique. And the first thing that we found, obviously the mouse is in the cage, you have to put your hand in occasionally to either drop something in or pick up the mouse. And it was very obvious from the onset when we put our hand in that we had quite a strong increase in activity from the PFC to the LH, as you can see here. This is area under the curve before and after, and this is a heat map showing individual trial responses. So this was an indication that this pathway is active in response to acute stress. We then also applied an air puff stress. Essentially, the mouse was in its cage. It was allowed to move around and acclimatize and become happy with the environment. And then we applied an air puff, and you can see in response to air puff, you had a dramatic and immediate increase in activity in this PFC to LH pathway. And again, here's the heat map showing individual trial responses. The final experiment that we did and that I'll talk about today is we introduced either an intruder mouse, which was not known to the experimental mouse, or we had a 3D printed mouse object, which is shown in the gray. And what you can see here in orange, this is the approach to the intruder, and you can see the activity of this PFC to LH pathway increased as mice approached the intruder. They have an interaction. And then, although it's still high, it eventually goes down over time as the experimental mouse assesses that the intruder is not posing any existential threat. And so what we can see is that the PFC to LH circuit is active in response to stress and threats, or in anticipation of a stress or a threat. But the activity wanes once a potential threat is assessed as safe. So together with this data and the activation and inhibition data suggests that the PFC to LH circuit assess acute threat and then suppresses feeding. And this is actually, this ability of acute stress to suppress feeding has been shown previously, but we are now showing that this novel circuit is involved. And of course, the reason why we started looking at this pathway was because of the modeling work done by Antonio and Kati, and it was suggested that there was less influence of this pathway in obese humans. And so we wanted to try and replicate that. And in order to do that, what we did is we applied another genetic approach where we can actually kill the pathway, remove the PFC neurons projecting to the LH. And that should should essentially mimic what's going on in the obese condition in humans, in which the pathway was predicted to be or was found. And what we can see is that cumulative high-fat diet food intake was increased when we removed this suppressive pathway. And body weight gain, now just on a chow diet, which is really interesting, mice gained more weight on a chow diet over time. You can see there's some fluctuation because we were doing other experiments on them at the time. But this is just increased body weight gain on a chow diet. Again, recapitulating what the human evidence predicted in the first place. So what we've done here is we've actually taken information from neuroimaging and modeling, and we have assessed the functionality, because obviously you can't test the functionality or causality of a pathway in humans. We've applied this in our preclinical models, and we've tested the functional consequences of this novel PFC to LH circuit. I should mention there is no information in the preclinical literature about this pathway in terms of feeding and body weight. So it's a really novel way. And I should also say we would have never sought this pathway or thought to activate or look at this pathway without the information from Antonio and Kati. I think it's interesting because rather than a mechanism that drives excessive consumption, we've actually identified a novel circuit that restrains feeding and sucrose seeking in response to potential threats and stress. And loss of this inhibitory restraint promotes weight gain. And the final point that I wanted to say was, I think this highlights the power of interdisciplinary collaboration. Again, we would have never looked at this pathway had it not been for the information that Antonio and Kati were able to produce. And so with that, I'd just like to acknowledge people that have helped, particularly Rachel Clark, that's done a lot of this work as a part of her PhD. Other members of the lab, of course, Antonio's lab and Kati, and then other members that have helped us establish the recording techniques and also the mouse vending machines. So thank you very much for listening.
Thank you very much, Zane, and thank you very much, Kati. Those were wonderful presentations. So I'm going to try, well, first of all, hello everyone, and I would remind everybody or encourage you to try to put some questions in the Q&A, and we will address them in a minute. I just wanted to do a really quick presentation to wrap up some of the points that Kati and Zane have been making, and also to showcase a couple of studies that we're about to start that are based on the knowledge that we have generated with this reverse translation approach. And I guess one of the things that I wanted to highlight is obviously we're doing this seminar in obesity organized by the Monash Addiction Research Center. And the reason for that is that there is this view out there that some forms of obesity or some symptoms of obesity resemble those of people with substance use disorders or addictive disorders. So I thought it would be interesting just to give you a little bit of a summary of what we think about what goes wrong in the brain in people that have addiction problems. And the current understanding is that addiction is linked to alterations in at least these 4 different systems. So on the one hand, you have the incentive salience or the reward system. And what happens here is that this system becomes very exclusive to everything that has to do with the drug or with the addictive activity. And the analogy would be that in this case, becomes really, really exclusive with everything that has to do with high energy food, calorie dense food, at the cost of other alternatives that could be healthier. Then you have alterations in a second system that is the one that regulates stress and negative emotions. And I think Zane's data on stress was really compelling in terms of showing how important it can be to drive food intake. And then you have a third system that is what we call executive functions or executive control, which is all about your goals and what you want to achieve and how to regulate and potentially control your behavior, especially that the behavior that is somehow incompatible with your goals. So for example, if you want to be healthy in the midterm, let's say that you cannot be eating McDonald's every single day because that would be incompatible with the goal. And I guess like it takes obviously effort and capability to try to control some of those behaviors to be able to regulate it. And then we have alterations in the decision-making system, which are probably best represented by the interaction between the other 3 systems. Because every time you're facing a choice between different options, what you do is you retrieve some information from your reward system to tell you what is important, what is not. You retrieve some information from your emotions and your current state. You retrieve information about your goals. And the brain integrates all that information and eventually makes an informed choice about what it thinks that or how we think we can better satisfy our needs at that point in time. And I think that that's the kind of choice that we face on an everyday basis when we're doing food choices. So oftentimes we are kind of in a dilemma between these 2 options. And this is an oversimplification, but it occurs like this oftentimes, in which you have some options that are immediately rewarding and they look much more appealing and reinforcing in the short term. And then you have other options that you know they're good for you or they are better for you in the long term, but maybe they don't look as instantly pleasing as the others. And we've been trying to model those type of choices using the stomachial system that Kathy was talking about, or as a high school student that emailed me the other day named it is my milkshake machine. That's what he told me. He told me, Professor, I know you have a milkshake machine. I would like to participate in one of your experiments. So essentially what we're doing is we are presenting people with choices between some beverages that are relatively low in calorie and high in fiber, and other choices that are really dense in terms of calories and have a good mix of carbohydrate and fat. And people do binary choices, and at the end of the day, what we do is whatever they choose, we give it to them. And in that way, we are being able to separate those 2 different stages of the decision-making process. A first stage, which is about considering the different options and thinking about the different options. And a second stage, that is, once you have made a choice, you get that choice and you kind of get the pleasure or get the sensations that are coming from that choice. And we have seen, for example, that different regions seem to play a more important role in one stage or the other. So, for example, we have seen that during the thinking process, there is an important contribution of the anterior cingulate cortex And during the consumption phase, we have very strong activation of the ventromedial prefrontal cortex. But the interesting thing is that when you analyze the relationship between body mass index and the activation of these ventromedial prefrontal region, which is kind of tracking the liking or the reward value of the options when you consume them, we have found, for example, that there is a negative association between BMI and VMPFC activation to the choices that people with obesity have made. So even though they selected those because they were the ones that they liked, the brain doesn't seem to be tracking the rewarding value. We are interpreting this as having blunted liking signals in the brain. And on the other hand, we have seen that the ventromedial prefrontal cortex of people with greater BMI becomes active when they have easy choices, when they are faced with choices in which the value of one of the choices is clearly much better than the value of the other choice. And kind of thinking about easy choices, I think about control, right? Like if something is easy and available, like if you want to really withhold the immediate tendency, you're going to have to exert some control. And this is one of the key exposures that Zane and Kathy were talking about. So I got this email the other day: feed your cravings in just a few clicks, right? You're just a few clicks away from all the pleasures you want. Including alcohol, by the way. So you have a good mix here of high calorie food and alcohol, which is another debate. And I was checking this email yesterday and I instantly got a reminder to order. So you have machine learning systems telling you what to do at each point in time. And then, like, one of the things that we want to understand is how obesity is associated with more or less difficulty to control those immediate responses. So we have designed this cognitive impulsivity suite, which is an online assessment tool to measure impulsivity. And we're going to be doing some studies to see how people with obesity and disordered eating make this type of impulsive choices. And the other thing we're about to start is a transcranial magnetic stimulation experiment, which is informed by the findings that Kathy and Zane were presenting today, and about the relevance of the medial prefrontal cortex in driving food choices and food cravings. So we're going to put our recruitment calls out there relatively soon. So keep an eye on that. And if you're interested, or you know people that could be interested, please email us. Thank you very much to everybody that has been involved. We also have an organization, the Addictive and Compulsive Eating Research Organization, that is interested in this debate on whether food addiction is an interesting concept and how we could explain some of the analogies between addiction and obesity. Yep, anything, if you're interested, just drop me an email or check our website, or keep in touch with Zane and Kathy as well. So that was it from me. And if I can stop sharing, and I know like we are a little bit short of time, but I would like to ask you a couple of questions, Kathy and Zane. And I think they're relevant to both of you. So both in Kathy's models and in Zane's experiments, you mostly have, let's say, input coming from cortical regions into subcortical and deep brain regions. But what about the other way around? What about communication between regions in the core of the brain, or even communication that is coming directly from the body into the brain? Like, would that be equally influential? Why is not showing up in some of the models?
Maybe I'll answer that initially from my point of view. Again, we looked at this input to the hypothalamus based on some of the work that you guys, some of the modeling that you predicted and showed us. But it's well known, I think, now that regions in the hypothalamus, such as hunger sensing neurons, they engage with centers outside of the hypothalamus. So they engage with reward centers. They engage with centers involving mood. And when you're hungry, you're actually having homeostatic input from the hypothalamus going to the reward centers, and you're potentiating the reward response. And that's interesting because there is anecdotal evidence that drug takers will fast before they take their drug of choice in order to get a greater high. So there is definitely information being passed from the hypothalamus to other parts of the brain. And of course, in terms of mood, well, when you're hungry, it's really important to dampen down your stress to make sure that you can explore your environment from an evolutionary context in order to find your food.
Yeah, and just from the neuroimaging perspective as well, like it's just because it doesn't show that there is this higher connectivity from the hypothalamus, for example, to cortical areas. It doesn't mean that they're not existent. It just means that the model emphasizes other areas or connections that are more driving in that case. And then what Zane also tipped on is that the hypothalamus actually integrates a lot of signals that coming, for example, what you also asked, Antonio, from the gut, for example, ghrelin, leptin, that are hormones that actually impact the hypothalamus and then the whole system as well. I think you're muted.
Yeah, you're muted, buddy.
Oh goodness, I still don't know how to use this in 2021. So the other thing, which obviously could be interesting for part of the audience, is how do you think your results could inform the debate around food addiction?
I'll let you take this one, Kati, first.
Yeah, it's an interesting one because as we know, it's a hard discussion at the moment. Is obesity or is overeating a food addiction, or can we claim it as an addiction? Because obviously there are similarities between drugs, but there are also a lot of dissimilarities. For example, we decide every day, like at least 3 times a day, what to eat. So I think that's still a tricky one, and it's, I guess, also splits the audience how we can put that field in terms of addiction, and we are actually also planning a new experiment to see whether we can make these comparisons. So I guess there are a lot of more empirical questions to ask if we actually can put it in that category or not. So my brief answer is I'm very careful to put it in that category.
And I would echo those sentiments as well, because I think there's evidence to be interpreted both ways. For example, when you're hungry, there are hunger sensing neurons in the hypothalamus that can directly increase the amount of dopamine released in the reward system. But those hunger neurons in obesity, they don't work as well. They're dampened down because they're not sensing hunger in the same way. So yeah, I think you have to be careful how we interpret the different studies. What is interesting, I think, is this drug called Contrave that I'm sure everybody's heard about. This is a drug that is FDA approved to suppress food intake, and it's a combination of naltrexone and bupropion, and essentially it's acting on reward centers in the brain to reduce food intake. And I think that's a really illuminating point and highlights where we should really be looking.
Thank you very much, guys. Okay, I'm gonna hand over back to Susie for the closure. Thank you very much. It was really exciting to hear about your work.
Great, thank you so much to all of our speakers, and I just found that fascinating, particularly the idea that they might be able to get personalized ideas about how to target people to individuals based on these studies. So thank you so much to all of our speakers. I just wanted to, before we go, to let you know about a couple of webinars that are coming up. So we have next month, our webinar will be on the socioeconomic lens of addiction. And so looking at some of these economic factors that drive addiction, particularly among disadvantaged groups. So we have 3 great speakers, Sonia Denou, Philip Mendez and Aaron Schlonsky in June. We also have, I'll let you know, our July webinar also looked in changing tack slightly, looking at cannabinoids and how they might help us address the opioid crisis. So this is our first international speaker coming for the webinar series. So we'll have Kelly Dunn joining us from the Johns Hopkins University, so Associate Professor Dunn, and we'll also have a panel discussion with our own Professor Rochelle Bookbinder and myself talking about potential role of cannabinoids in the opioid space. So 2 webinars to look forward to. I just really like to extend my thanks again to today's 3 fantastic speakers, really illuminating a very complex and exciting area that we haven't looked at a lot in our webinar series before. So thank you again, Dr. Voigt, Professor Andrews and Professor Verdejo-Garcia for joining us today. And thank you to everyone who's joined in the audience.